Welcome back to my channel Knowledge Chronicle.
Have you ever wondered why, despite countless generations, some traits in a population remain unchanged, while others evolve dramatically? What invisible law keeps gene frequencies stable, or suddenly shifts them, giving rise to new species? Welcome to Knowledge Chronicle. Today, we're decoding the Hardy-Weinberg Principle, a powerful mathematical model that reveals how evolution works behind the scenes. Stay with us till the end because this isn't just theory, it's the heartbeat of population genetics. Let's start with the basics. The Hardy-Weinberg Principle, proposed independently by G.H. Hardy, a British mathematician, and Wilhelm Weinberg, a German physician in 1908, is a foundational concept in population genetics. It states that, in a large, randomly mating population, where no external forces act, the frequency of alleles and genotypes will remain constant from generation to generation. In simple terms, if no evolutionary forces are acting, the population is in genetic equilibrium. But what is this allele frequency? Alleles are different versions of a gene. For example, the gene for eye color may have alleles for brown, blue, or green eyes. If in a population the frequency of allele A dominant is P and the frequency of allele A recessive is Q, then the total frequency must be P plus Q equals 100% of alleles. Now, for genotypes, combinations of alleles AA individuals equal P squared, AA individuals equal 2PQ, and AA individuals equal Q squared. So P squared plus 2PQ plus Q squared equals 1. Alright, so this is a binomial expansion of the expression P plus Q, all squared. Now let's try to understand this concept a bit better with an example. Okay, imagine, if you will, a population of 1,000 people. If P equals 0.7, which means 70% allele A, then Q equals 0.3, which is 30% allele A. Then AA equals P squared, which is 0.49 or 49%, translating to 490 individuals. AA equals 2PQ, which is 0.42 or 42%, translating to 420 individuals. And finally, AA equals Q squared, which is 0.09 or 9%, translating to 90 individuals. These proportions will remain constant if the population is in Hardy-Weinberg equilibrium. What is genetic equilibrium? The term genetic equilibrium simply means allele frequencies do not change over time. It's like a perfectly balanced scale. Unless you disturb it, it stays unchanged. This is crucial because any change in allele frequency indicates that evolution is happening. So, how do we know evolution is occurring? By measuring real-life allele frequencies and comparing them with expected Hardy-Weinberg values. If they differ, there's your evolutionary signal. When does Hardy-Weinberg not work? The principle works only under ideal conditions, but nature is rarely ideal. There are 5 major factors that disturb genetic equilibrium. When individuals migrate from one population to another, they carry their genes with them. Example: if dark-skinned beetles move to a population of light-skinned beetles, they introduce new alleles. This is a random change in allele frequency, especially significant in small populations. Example: imagine a few red flowers blown away by a storm. Suddenly, the gene for red flowers becomes rare. A special case of genetic drift is the founder effect, a fascinating concept in evolutionary biology. When a small group breaks away to form a new colony, they carry only a sample of the gene pool. It's like a tiny snapshot of the original population's genetic diversity. Over time, this isolated population may evolve very differently from the original group. It's quite intriguing how these changes can lead to unique evolutionary paths. Mutations are sudden changes in DNA. Even a small mutation can introduce a new allele, altering frequencies. For example, a mutation may cause a new pigment in moths. If this new pigment offers a survival advantage, it tends to spread within the population. Let's delve into genetic recombination. Genetic recombination occurs during sexual reproduction, especially in meiosis. During this process, crossing over and independent assortment mix alleles, creating genetic diversity. So, this adds variety without changing allele frequencies directly, but it does affect how traits are combined. It's like mixing up the deck of cards, you know? Alright, let's dive into the fascinating world of natural selection. Now, this is the most powerful force in evolution. It's like the ultimate game changer, really. Over time, allele frequencies shift towards survival advantage. Example: during the Industrial Revolution, dark-colored moths survived better on soot-covered trees. Tidal outcomes of natural selection. Natural selection can lead to three possible outcomes in populations. First, stabilizing selection. Stabilizing selection favors average traits and eliminates extremes. An example of stabilizing selection is human birth weight. Babies that are too small or too large have higher mortality rates. It's the average-sized babies that tend to survive best. So let's move on to directional selection. Directional selection favors one extreme of a trait within a population. For example, in drought years, birds with longer beaks might survive better by accessing deep seeds. Third, let's talk about disruptive selection. It favors both extremes, not the average. So, here's an example. In a habitat with black and white rocks, both black and white rabbits survive, but gray ones are easy targets. These types of selection change gene frequencies, driving evolution forward. Now let's delve into experimental proof and speciation. Microbial experiments give us direct proof. In labs, scientists expose bacteria to antibiotics. Some bacteria already have mutations making them resistant. These bacteria survive, multiply, and eventually, all bacteria become resistant. This is selection of pre-existing advantageous mutations. Over generations, as changes accumulate, we get speciation, the formation of new species. Let's bring it all together. The Hardy-Weinberg Principle is like a baseline, a control condition. It tells us what a population looks like if nothing changes. Any deviation from the expected frequencies reveals the presence of evolutionary forces. Whether it's migration, drift, mutation, recombination, or natural selection, this principle is not just for theory. It's used in studying inherited diseases, predicting carrier frequencies, understanding population structure, and even conservation biology. It's the mathematical fingerprint of evolution. If you enjoyed this journey through the invisible world of genes and evolution, don't forget, your one like, share, and subscribe will help us create more valuable content. This is Knowledge Chronicle, where science meets curiosity. Until next time, keep asking, keep exploring.